like to thank the organizing committee for giving me this opportunity to talk, and for all of you for welcoming me into this community, and for the Alliance for supporting some of our research. I do have one disclosure. I'm a yeast geneticist, so please lower your expectations for this talk. So I'm going to start off with just sort of this overview of drug development, but then I'm going to do a tangent into my background that brings me into this specific talk. So generally, drugs are developed by first identifying some kind of target, generating an assay for it, and then carrying out some sort of high throughput screen, testing anywhere from thousands to millions of compounds, finding active compounds, and then going through medicinal chemistry to modify them to make them more suitable for drug use, testing them in various models, and finally getting to clinical trials. So where do I come in with all this? So my background as a yeast geneticist is that I started in the field finding a biological process of interest that would be shared between yeast and more medically relevant organisms such as humans, and then using a mutational analysis looking for mutations that affected that biological process to study how it works. So what I like to say is I thrive in the absence of knowledge, and therefore I started to study how fission yeast senses glucose, this particular sugar, in its environment and regulates gene expression in response to that. What I learned was that mutations that prevented the sensing of glucose identified the cyclic AMP signaling pathway of fission yeast. And so my lab at Boston College cloned the genes of this pathway, and it looks very much like a hormone signaling pathway in humans. So where we come in is that about 15 years ago, we started, rather than simply studying the process in yeast, replacing the enzymes that produce or hydrolyze cyclic nucleotides in yeast with those of mammals or pathogens so that we could do small molecule screening. So we've done screens of anywhere up to about 240,000 compounds for inhibitors of the enzymes that break down the cyclic AMP. And then in 2015, I met Mike Collins at a cyclic AMP signaling conference where he gave this wonderful talk about GNAS and this need, and McCune-Albright syndrome, and the need to identify compounds that might be able to be used to treat it. And my thought was we had been working on the side of compounds that elevate cyclic nucleotides, but I knew we could also look for compounds that would reduce it. So I went up to him during the coffee break afterwards. He was very welcoming and gave me clones of wild-type and mutationally activated GNAS, and then I sought out clones of the adenylyl cyclases, of which in humans there are 10 different genes, 9 of which respond to GNAS, and a 10th which does not. And so we constructed yeast strains that expressed each of the 10 different mammalian adenylyl cyclases. So I'm going to go very quickly through this. You know that in McCune-Albright syndrome, GNAS is mutationally activated, so it's stuck in the on position, and it works by stimulating these transmembrane adenylyl cyclases. So this cartoon doesn't actually do a good job of depicting what a transmembrane adenylyl cyclase looks like, but I'll get to that later in the talk. So now we have increased expression of or production of cyclic AMP, and so again, we developed a screen where we would be able to screen thousands of compounds for ones that could lower cyclic AMP production. And so I did that. Actually, remarkably, I personally carried out the screen at NCATS. This is the National Center for Advancing Translational Sciences. So during a sabbatical, I went there and worked on optimizing this screen. Then with support from NIH, I came back and carried out 2 additional screens of this collection of 125,000 compounds, found 5 that were structurally related and could, in fact, lower cyclic AMP production by both our yeast cells that specifically express mammalian adenylyl cyclases that were responding to mutationally activated GNAS, and also in HEK cells, human embryonic kidney cells. And through a collaboration with Fernando Fierro, he showed that they could also reduce the cyclic AMP levels in osteoblasts. And that work was also supported by a $1 million bike ride grant. Other support we had gotten from $1 million bike ride grant gave us the ability to expand from those 5 compounds to a collection of 41 compounds and identify that 12 of them were active. Now here's where we run into a problem. Another group at Purdue had found the same structure in a collection of compounds using HEK cells that were genetically engineered to express adenylyl cyclase 1. So the GNAS responding cyclases are cyclases 1 through 9. So Val Watts' lab had genetically engineered HEK cells to specifically express adenylyl cyclase 1, but instead of activating them with GNAS, he was stimulating them with a calcium ionophore. When it came time for him to look at the specific role of, or specific ability of the compounds to inhibit the cyclase using in vitro enzyme assays, because that's the gold standard for chemists, they didn't work. So that left him to think that maybe these things were working through calcium signaling, but that would not work in our system. So that didn't make any sense, that he failed to see this in vitro enzyme assay result, and yet we both came across the same set of compounds, or overlapping sets of compounds. So as I said here, he concluded that it might be due to calcium signaling because he had been burned in the past with a very similar story. And so he kind of lost interest in these, and I just didn't think there's any way this could have been calcium signaling. Okay, so the enzymes we were working are not regulated by calcium. And so here we have this dilemma. It's like, do these compounds actually work directly on adenylyl cyclases, which is why I have in my title putative adenylyl cyclase inhibitors, or might they be working through some other mechanism? And so this leaves us 3 possible mechanisms of action. They could prevent GNAS from activating the cyclase. They could inhibit the cyclase independent of GNAS function, or they could act on a process that's required for adenylyl cyclase activity. Now, why is this a problem? Because if we want to do this step here in the blue box of getting a medicinal chemist to start making derivatives of these compounds so that we could get more structure-activity relationships, find more potent enzymes, find compounds that are more suitable for use as a drug, we have to, they're going to want to see in vitro data. They want to be guided by how potent it is in an enzymatic reaction, and we can't do that. And so and if we want to get funding for that work, it's going to be a study section of chemists who also want to see in vitro data. So we're stuck at this point of saying, like, what are these compounds doing? Is there any way in the absence of an in vitro enzyme assay to convince these people that we actually do have compounds that are directly working on the cyclases? So in this experiment, we address the question of whether GNAS needs to be present or not for our compounds to have an impact. So on the top panel, you see a yeast strain that is expressing the yeast adenylyl cyclase, and the collection of dots in these 3 panels along the top indicate the fluorescent signal in these cells. The fact that, and DMSO on the far left, is the vehicle control. Here are 2 of our compounds in the middle and far right, and the fact that there's no shift in that signal means that the compounds are having no impact on a strain that has the yeast enzyme. The next 2 sets of panels have either adenyl cyclase 2 or 4, And in both cases, you see a rightward shift indicating that the compounds are inhibiting cyclic AMP production. Whether it's direct or not, we still can't conclude, but it's not that you need GNAS present to see the impact. And we've also seen this with strains that express cyclases 7 and 9. Okay, so now we're going to finally take a look at what these adenylyl cyclases really look like that are responding to GNAS. And what they have are 12 transmembrane passes, so they go in and out of the peripheral membrane of the cell 6 times. They have a cytoplasmic domain called C1, then they go back through the membrane 6 more times in the second cytoplasmic domain, and the 2 cytoplasmic domains come together to make the active site. The second cytoplasmic domain is where GNAS inserts a helix when it becomes activated to somehow stimulate the activity, and I need to talk more with real cyclase experts to understand better what the consequence of that interaction is. Okay, so here was our thinking. There are a couple ways that we might be able to distinguish between a direct effect or an indirect effect. And the first one is, do we see patterns of inhibition by these various active compounds that would suggest that it's a direct effect? And my thinking is that if we can see very distinct patterns, like compound 51, if you look at this little panel there, compound 51, it seems to be a much better inhibitor of cyclase 7 than, well, wait, I'm saying it the other way around. The lower it is, the more inhibition we're seeing. So it's not a very good inhibitor. 51 is not a very good inhibitor of cyclase 7, whereas if you look at 67, it's probably better on cyclase 7 than it is on cyclases 5 and 9. So this kind of specificity might suggest directness. However, the way we were doing it, we don't have statistically significant data. So we're going back to this approach, but doing it more in dose response curves that will hopefully give us more specificity. The other approach, though, is to modify the enzyme and look at the impact of modifying the enzyme on the response to the compounds. So we have constructed a chimeric enzyme. So chimeric means it's composed of 2 different portions, most of which is adenylyl cyclase 4, except that the C2 domain comes from adenylyl cyclase 6. Now, my thinking at the time was it might be able to tell us whether the compound binds to the C1 domain or the C2 domain. And sometimes the best data, and this is wildly preliminary because it's about 1 week old and has not been reproduced yet, the best data are the ones you don't expect. And here's what happened. So we do this dose response curve, because that's now how we're doing it, with a strain that expresses adenylyl cyclase 4 versus 6 versus the chimera. And what we see was completely unexpected, that the chimera appears to be hypersensitive to inhibition by these compounds. So again, I was thinking, oh, it's going to mimic one or the other, not that we're going to see a completely new response. But what that did was give us a new model for what's going on. And it suggests the idea that what the compound actually is doing is going between the interface between the 2 chimeric, or between the 2 cytoplasmic domains to interfere with activity. That would explain why it works on the basal enzyme. That would also potentially explain why it doesn't work in the in vitro enzyme assay. Because now if you propose that part of the role of the membrane is to limit the interaction between C1 and C2 by some sort of steric means, if you make a membrane extract, you take that away. If you have there's a synthetic form of the enzyme where people have simply produced the C1 domain and produced the C2 domain and allow them to dimerize independent of a cell, that would also potentially take away that interface for the compounds to work. So that's where we are now. So very quickly, what we've seen is that, again, mutations in GNAS elevate production of cyclic AMP by these transmembrane cyclases. We have a collection of structurally related compounds that reduce cyclic AMP production, specifically in the yeast cells that make the transmembrane cyclases and GNAS, also if they make the transmembrane cyclases without GNAS. They don't work in the biochemical reactions, but they do show these characteristics that would suggest that they are working potentially by blocking the interaction between C1 and C2. In the future, we are about to construct some new mutants in adenylyl cyclase 4, where we just change one amino acid in a possible binding site on either the C1 side or the C2 side, with the idea that that might confer resistance to the compounds. I think that if that were the case, that'd be very strong evidence for direct interaction. And thus, our hope is that this sort of work could eventually get us to a point where a medicinal chemist is willing to come on board and a funding agency might be willing to support such medicinal chemistry. So I just want to quickly acknowledge people in my lab who did this work, our collaborators at NIH and NCATS and Purdue, people who gave us the adenylyl cyclase clones, Dr. Watts, DeSauer, and Cooper, funding from NIH and from $1 Million Bike Rides and from the Reeser Lectureship Fund. And thank you for your attention. I'm happy to take questions depending on time.